Hello, everyone. My name is Shen from Texas A&M University. Today, I'm talking about de novo protein design for novel folds with guided and conditional Wasserstein GAN. This is a joint work with my PhD students, Mustafa, Shen, and Yue. So our group is interested in modeling biosystems, especially the parts, proteins, DNAs, and RNAs. We model or design these molecules or their interactions so that we can forward and reverse engineer biosystems. So one kind of problem is a forward problem that we start with a molecular identity and predict its function, for instance, starting with the protein sequence and predict its structure or function. But the focus of today is the inverse problem, that we start with the desired property or function and design or predict the protein identity. For instance, protein design is the inverse problem to protein folding. So you may wonder, algorithmically, is there any special need for inverse problem? Sounds like I could have just applied the forward problems algorithms to all the possible candidates X, and then I just figure out which X has the best Y. Well, theoretically, it's possible, but you have to consider that the space of these molecules, X, is huge. So applying the discriminant models in the forward problem to the inverse problem is finding needles in a haystack. So rather, you may want to apply or develop generative models that can directly model the distribution of the sequences or molecules X given the desired property Y. We have seen a lot of the advances of such methods in drug discovery lately. So our problem is, can we make proteins of any desired fold or eventually shape and function? So this problem is a little bit different and has its own unique challenges compared to the drug discovery or molecular design there. One thing is that the sequence fold relationship is very complicated. It's both convergent and divergent evolution. Sometimes we see very dissimilar sequences of the same fold. And meantime, our desired property Y is in the discrete space, so it's categorical. So we are actually designing sequences with desired Y value or fold classes that have never been observed in the training set. So these are the unique challenges to our problems. So the approach we take is a data-driven approach because we want to answer how adequate are current protein data in revealing the sequence structure relationship, such that new folds can be designed accordingly. So this is a critical test to the so-called learned structure sequence relationship. So what data do we have then? So we do have protein sequences that are labeled with the folds. Meanwhile, we do have much more data that are not labeled. So they are going to be utilized in the semi-supervised learning setting. And the model we developed is a semi-supervised, guided, and conditional Wasserstein GAN. So it's again with generator and discriminator playing a game. And it's a Wasserstein GAN because the Wasserstein distance is replacing the KL divergence, not only for the training stability, but also for the design's diversity, as you will see later on in the results. And it's a conditional GAN because we are conditioning the generator, the sequence generator, on the desired property, in this case, the fold. So we actually use the pairwise distances between the fold pairs and design a generalizable low-dimensional fold space. So based on some of the folds, we can represent the other folds. And lastly, it's a guided GAN. Compared to the regular GAN, you see that in addition to generator and discriminator, we have a third network that's called an oracle. It provides a quick feedback that can help guide the generator. So it actually have to predict folds for thousands of sequences within a second. So we had to do some revision of a state-of-the-art method. So after that, of course, this model is trained, starting with unpaired sequences, and then trained with the label data after that. So how good are the designs? The first question we ask is, are these proteins stable? So using the predicted instability index, we see that they resemble the natural sequences and much more stable compared to random. The next question is, what about the other biophysical properties? We checked the gravity, isoelectric point, normalized molecular weight, aromaticity, and they all resemble the natural sequences in these properties and very differ from the random sequences. So the next question is, are these designs functionally specific? So we predicted the geo terms of these designs and then compared the geo terms to the known geo terms of target folds as well as the off-target folds. So by comparing these geo terms, we found out that not only for molecular function, but also for biological process, our designs are more resembling the target folds in the function compared to the off-targets with statistical significance. So lastly, remember, we are designing sequences for a desired fold. So how good are these? So here we're just looking at one case study. The others can be found in the report. This desired fold is not even in the basis folds that we use to construct the fold representation. So the question is, how good are they? So we use the Rosetta to predict the structures and calculate the TM scores between the designed sequences and the ground truth. So we found that the fold accuracy is comparable or slightly better than the competing method called conditional VAE. And the best design has a TM score around 0.46, which is very close to 0.5, the threshold to tell the same identical fold. More importantly, our designs are much more diverse among each other, and they are much more novel compared to the baseline. So lastly, we integrated a data-driven and principle-driven Rosetta. So if we let GCW again serve the seed designs in Rosetta, the success can be doubled. And then if we use the profiles of GCW again to reduce the sequence space, when we reduce that by 10 to 76, when Rosetta cannot find successful designs, actually GCW again can still help Rosetta find a lot of successes. So in the summary, we have described a generative model for an inverse problem. The question is, how do we directly design a molecule of desired property? In this case, a structural fold. So we have designed a Wasserstein game that is semi-supervised, guided by an oracle, and conditioned on the property embedding. The designs are biophysically, functionally, and structurally meaningful based on our predictions. At the same time, they are more diverse and sometimes more novel compared to a competing method. And more importantly, at the end, we show that integration of data-driven and principle-driven methods will boost the success of the Novo protein design. With this, I will end today's talk by thanking my group members and collaborators and funding resources, and thank you all for your attention. I'll be happy to take any questions. Thank you.